Hi. The highlighted topics in this session are heartbeat, cardiac cycle, blood pressure, sphygmomanometer, stethoscope, blood group, and lymphatic system. Let's discuss the topics one by one.
Our first topic is heartbeat. So let us see what is heartbeat. One complete contraction and relaxation of the atrium and ventricles of the heart constitute heartbeat. So what is heartbeat? One complete contraction and relaxation of the atrium and ventricles of the heart constitute heartbeat. The heart normally beats 72 to 75 times per minute. So per minute 72 to 75 times. Yes. So what is heartbeat and the heartbeat times? How many times? Now next we have to see neurogenic and myogenic heartbeat. What is neurogenic heartbeat and what is myogenic heartbeat? First we have to see neurogenic heartbeat. Neurogenic heartbeat is initiated by the nerve impulse caused from the nerve ganglion situated near the heart. So the heartbeat is initiated by the nerve impulse. So it is known as neurogenic heartbeat. The heartbeat is initiated by the nerve impulse caused from a nerve ganglion situated near the heart. Example, annelids and arthropods. So these are the examples. The neurogenic heartbeat is initiated by the nerve impulse. Now myogenic heartbeat. Myogenic heartbeat is initiated by the specialized group of modified heart muscle fibers. That is a specialized group of muscles. So it is known as myogenic heartbeat. Here initiated by the nerve impulse. Here initiated by the specialized group of heart muscles. Yes, example mollusca and vertebrates. Human beings also myogenic heartbeat. Yes. Now next topic, initiation and conduction of heartbeat. Initiation and conduction, how the heartbeat initiated and how it is conducted. That only we have to see now. The human heart is myogenic in nature. Human heart is myogenic in nature. Now we have seen what is myogenic heartbeat. Yes, so it is initiated by the specialized group of heart muscles. So human heart is myogenic in nature. Contraction is initiated by the specialized portion of the heart muscle, the sinoatrial node. So the specialized portion of the heart muscle, that is SA node, sinoatrial node, which is situated in the wall of the right atrium near the opening of the superior vena cava. Now see this diagram. So here right atrium, left atrium, right ventricle and left ventricle. Yes, now we have to see where is the SA node. See here in this top area near to the right atrium. Yes, right side atrium and near to the superior opening, near the opening of the superior vena cava. This is a superior vena cava. So opening of the superior vena cava, this SA node is situated. This is the SA node. Okay, sinoatrial node. So this is a special type of muscle, heart muscle. Yes, this SA node is broader at the top and tapering below. So this region, it is the broader region and below that is a tapering. Yes, it is made up of thin fibers. This SA node is made up of thin fibers. So where it is situated? SA node, where it is situated? Which is situated in the wall of the right atrium, the right side right atrium, as well as near the opening of the superior vena cava. In that area, SA node is situated. Understand? Now, next one. The sinoatrial node acts as the pacemaker of the heart because it is capable of initiating impulse which can stimulate the heart muscles to contract. It acts as the pacemaker of the heart. Do you know what is pacemaker? Pacemaker is the device. Yes, so when if the heart is not functioning properly, immediately the doctors through the surgical method, the doctors can operate in that particular area and insert the pacemaker. So instead of sinoatrial node, they have to insert the pacemaker in this area. This is a sinoatrial node. So here doctors have to insert the pacemaker. So that acts as a sinoatrial node. So that is the artificial device. Understand? Yes, so this sinoatrial node acts as the pacemaker of the heart because it is capable of initiating impulse which can stimulate the heart muscles to contract. So this SA node only initiating the impulse which can stimulate the heart muscles to contract. Understand? Now, the impulse from the sinoatrial node spreads as a wave of contraction over the right and left atrial wall, pushing the blood through atrioventricular valves into the ventricles. Now, because of the impulse, one wave of contraction is present in this area. So, because of the wave of contraction, the blood pushes through the tricuspid and bicuspid valve. Yes, so the blood enters into the ventricles. Understand now? So because of the wave of contraction, the blood pushes into the bicuspid and tricuspid valves and enters into the ventricles. Now, the wave of contraction from the sinoatrial node reaches the atrioventricular valve. Now, this wave of contraction that impulses enters into the AV node. This region, this is the AV node, atrioventricular node. So that impulse enters into the AV node, then via atrioventricular bundle and the Purkinje fibers, it enters. Understood? So through the first the impulses or that wave of contraction enters into the atrioventricular node. After that, via atrioventricular bundle and the Purkinje fibers, these impulses enters into the ventricular muscles, so that the ventricular muscles contract. Understood, children? Yes. So first the SA node, sinoatrial node produce the impulse. That impulse enters into the which region? AV node, atrioventricular node via bundle of branches. This is a bundle of branches, Purkinje fibers through this area. It enters into the ventricular muscles, ventricles, so that this ventricles contracts. So in this manner, initiation and conduction of heartbeat takes place in human beings. I hope all understood. Okay, now we have to see the exercise. Exercise number 1. First question is define heartbeats. Define heartbeat. Second question, sinoatrial node acts as pacemaker of the heart. Why? So first question answer, this is the first answer. One complete contraction and relaxation of the atrium and the ventricles of the heart constitute heartbeat. Then this normal heartbeat also have to write. Then second, the sinoatrial node acts as the pacemaker of the heart. Why? Why? Sinoatrial node is capable of initiating impulse which can stimulate the heart muscles to contract. So it is known as it acts as a pacemaker of the heart. Now we are going to see pulse. So what is pulse? When the heart beats, the blood forced into the arteries. The expansion of the artery every time the blood is forced into it. So it is known as pulse. So what is pulse? When the heart beats, the blood forced into the arteries. The blood forced into the arteries. The expansion of the artery every time the blood is forced into it. So this is called pulse. So the normal pulse rate is 70 to 90 per minute. 70 to 90 per minute. So how to calculate the pulse? That only we are going to see. So that matter only running at the background of the screen also. Yes, so how to calculate? Just we have to see. First you have to take 2 fingers, not your thumb. Remaining 2 fingers have to take and place it near to the wrist area. Place it near to the wrist. Just slightly press it. So just calculate the pulse rate. First take it to 15 seconds, then multiply the rate into 4 or 30 seconds you wait and multiply into 2. So you can get one answer, that is your normal pulse rate. So through that you can identify your pulse rate also. Can you calculate? Right. Now we are going to discuss about cardiac cycle. So first let us see what is cardiac cycle. The sequence of events occurring from the beginning to the completion of one heartbeat is called cardiac cycle. One heartbeat. Already you have studied about heartbeat. Yes. So here the beginning to the completion of one heartbeat, beginning to the end of one heartbeat is called cardiac cycle. So what is cardiac cycle? The sequence of events occurring from the beginning to the completion of one heartbeat is called cardiac cycle. Then during cardiac cycle, blood flows through the chambers of the heart in a specific direction that we all know. Yes, when the blood flows, it flows in a particular direction. While flowing oxygenated blood as well as deoxygenated blood can't mix. Understood? It won't mix. In a particular way only it can flows. Yes. Then each cardiac cycle lasts about 0.8 seconds. The timing also there. So each cardiac cycle lasts about 0.8 seconds. The events during a single cardiac cycle involves three events. Yes. Number one, first event is atrial systole. The first event is atrial systole. Second event is ventricular systole. And third one is ventricular diastole. Third one is ventricular diastole. Now you see here, what is systole? Systole meaning contraction. Yes. So atrial, that is a atria or auricles. So contraction of auricles, that is atrial systole. Contraction of auricles are known as atrial systole. The timing of atrial systole is 0.1 second. Understood? Then second one, ventricular systole. Ventricular systole means contraction of ventricles. The timing is 0.3 seconds. Number 3, ventricular diastole, that is relaxation. Diastole meaning relaxation. So relaxation of ventricles, that is known as ventricular diastole. Timing 0.4 seconds. Understood? So these are the three events. Now you see this diagram. This diagram represents a single cardiac cycle. So the timing, a single cardiac cycle lasts 0.8 seconds. So here A indicates atrial systole and B indicates ventricular systole and C indicates ventricular diastole. Yes. So here atrial systole, the timing is 0.1 second. Number 2, ventricular systole, timing 0.3 seconds. 3, ventricular diastole, timing is 0.4 seconds. Have you all understood? Yes. Now see the questions. First one, what is cardiac cycle? That is a 2-mark question. So answer, the sequence of events occurring from the beginning to the completion of one heartbeat is called cardiac cycle. That is the definition. Second question is, name the events involved in a single cardiac cycle. That is the second question. So that answer is atrial systole, ventricular diastole. This contraction of auricles, the timing, everything have to write. That is a 2-mark question. Have you all understood? Yes. Moving on to the next topic. Students, now we are going to see one interesting topic, that is heart sounds. Do you know what sound is produced by our heart? Yes, correct. That is lub and dub. The sound is lub and dub. How it is produced, do you know? We have to see that only now. The sounds of a normal heartbeat are known as lub and dub. So the sounds are known as lub and dub and are caused by blood pushing on the walls of the heart. How it is produced? by blood pushing on the walls of the heart. When the blood flows through the heart or through the arteries and veins, at that time blood pushing on the walls, so that heart to produce sound. That sound is known as lub and dub. The lub sound comes first in the heartbeat and is the longer of the two heart sounds. So first sound is the lub sound, that is the longest duration. Second sound is dub sound, that is shorter duration. Now through the diagram we can see how the sound is produced. The first heart sound lub occurs when atrioventricular valves closes. When atrioventricular valves close, the lub sound is produced. See here, this is the atrioventricular valves. This one and this one. These are atrioventricular valves. Atrioventricular valves means tricuspid valve and the bicuspid valve. Tricuspid and bicuspid valves are known as atrioventricular valves. So when the valves closes, the heart to produce what sound? Lub sound. Yes, tricuspid and bicuspid valves close. At that time the heart to produce lub sound. Now second one. Second heart sound dub occurs when semilunar valves close. When the semilunar valves close, the heart to produce dub sound. So this is first semilunar valve, this is a second semilunar valve. So when these valves close, the heart to produce dub sound. So these are the two sounds, lub sound and dub sound. Now, are you ready to hear the heart sound? Yes? Okay. Did you hear the sound? Yes. Now see here the first sound. Which sound you heard the first time? That is a lub sound. It is due to the closure of tricuspid and bicuspid valves. So closure of tricuspid and bicuspid valves, you heard the first sound, that is lub sound. Second sound is due to the closure of semilunar valves. Second sound that is due to the closure of semilunar valves, you heard, is it? That is the dub sound. So lub and dub sound, the heart to produce. Now one heartbeat is equal to a lub plus a dub sound. So one heartbeat is equal to a lub sound and a dub sound. Have you all understood? Dear students, now we are going to see one important topic, that is blood pressure. So in this topic we can see what is blood pressure, then what are the two readings, systolic pressure and diastolic pressure, hypertension, hypotension. So these are the terms we are going to discuss in this topic through the video. First we can see understanding blood pressure. When your heart pumps, it carries oxygen, blood, nutrients through the arteries in your body. When the heart pumps, it carries oxygen, blood, nutrients through the arteries in your body. That we know. Yes. So now we have to see what is blood pressure. What is blood pressure? Blood pressure is the force of blood against your artery walls. It is the force of blood against your artery walls, that is known as blood pressure. Yes. Now we can see the readings. So there are two readings which measure blood pressure. So in the blood pressure there are two readings which measure blood pressure. So the two readings we can see one by one. So the first reading is systolic pressure. Systolic pressure. Systolic pressure is the higher number. Systolic pressure is the higher number. It measures the force of blood being pushed around your body. It is higher in number. It measures the force of blood being pushed around your body. that is systolic BP or systolic pressure. When your heart contracts, this is the main point. When your heart contracts, at that time only systolic reading we can find. Then next one is diastolic pressure. So diastolic pressure is the lower number. Systolic pressure is the higher number, but diastolic pressure is the lower number. It measures when the heart relaxes between beats. Okay, relaxes when the heart relaxes between beats. So systolic pressure when the heart contracts, but here when the heart relaxes between beats. Yes. Now we have to see high blood pressure. Yes, high blood pressure. High blood pressure is when your blood pressure is always higher than it should be. So above the normal level, always higher than it should be. That is high BP or higher hypertension. This is also known as hypertension and is usually diagnosed. So the high BP or systolic pressure, that is known as hypertension and is usually diagnosed. Yes, when your levels are always above 140/90 mm of mercury. So here the normal blood pressure is 120/80. In your book 120/80 mm of mercury is given, but here 140/90 mm of mercury is there, but both correct only. So this also normal BP only. Yes, so when your levels are always above 140/90 mm of mercury, that condition is known as hypertension. Yes, hypertension above the normal level. Okay, next, high blood pressure makes your arteries less stretchy. So high blood pressure, so if the BP is high, it makes our arteries less stretchy and damages the walls of our arteries. It damages the walls of the arteries. Okay, high BP means it damages the walls of arteries, and this makes it easier for fatty deposits to build up. So because of that, easily fat can deposit in the arteries and the veins. See here, this is the fat deposition. So this is the walls of the artery, this one and this one, this is the walls of the artery, and here that is a fat deposition. So only little gap is there. So in this way we can see that is a full gap. So here little gap. So easily the blood cannot flow through this way. Why? Because of the fat deposition. And limits the circulation of blood and oxygen. So the oxygen and the blood cannot flow properly. So it limits the circulation of blood and oxygen. It cannot flow in this way properly. Yes. This can lead to a heart attack or stroke. So because of that fat deposition, this can lead to a heart attack or stroke. So that condition, so the person may get heart attack or stroke because of the fat deposition. Now, high blood pressure typically has no symptoms. Maximum the person cannot show any symptoms. Yes, so that easily cannot identify. Understood? If left untreated, it can lead to heart and circulatory diseases. So from the beginning itself, if we find, we can go to hospital and we can take the treatment. Is it? So we can get relief from that particular problem. Or else, if left untreated, it can lead to heart and circulatory diseases. At the end, last moment when we find, we cannot do anything, is it? So heart problems, heart attack, stroke or circulatory diseases the person may get. So anyone can have high blood pressure. It can be caused by eating too much of salt food. So the person is eating too much of salt food and drinking too much of alcohol, then overweight. So these are the causes for the hypertension or not being active enough. So the person always in the non-active condition. So take control of your blood pressure. So when we are taking care of our blood pressure, what we have to do, just we have to reduce the salt content, then avoid drinking alcohol, then reduce the weight, so we can control of our blood pressure. Yes, then the decrease in blood pressure, that is known as what? Hypotension. Increase in blood pressure is known as hypertension. Decrease in blood pressure is known as hypotension. Normal blood pressure is 120/80 mm of mercury. That is a normal. In book that level is given, but in this video we saw 140/90 mm of mercury. Anyway, both correct. Students, what's this? Is this a tube? No. This is an instrument, is it? The name of the instrument is stethoscope. Who else are using this stethoscope? Are you using? No, is it? We are not using this stethoscope. Then doctors are using. Is it? Doctors are using the stethoscope. See this stethoscope? Here different parts also present. Is it? Different parts are there. See, first one is ear tips. This is ear tips, and this tube, this is the aral tube. Then headset. This is diaphragm. This is tubing. Then this is bell and chest piece. So these are the parts also present in the stethoscope. Do you know these parts already? No, now only we are seeing this. Yes? Okay. Now we can see the stethoscope. What's the use of this stethoscope? A stethoscope is used to detect the sound produced by the internal organs of human body. Internal organs. It is an instrument. It is used to detect the sound produced by the internal organs of human body. Then the heart sound is heard by placing the stethoscope on the chest region. So when we go to hospital, doctors used to use this stethoscope. Is it? Then in which place they are keeping the stethoscope? Near to the chest region. Yes? Okay. It is a useful diagnostic tool. Identify and localize health problems and diagnose disease. So two uses. What are the two uses? First one is localize health problems. So this stethoscope is used to localize health problems, and next one is diagnose disease. So these are the two uses. It is used to localize health problems and diagnose disease. The modern electronic stethoscopes are high accurate instruments. Nowadays we are in the modern world, so different varieties of electronic stethoscopes also, they are new modern. So that stethoscopes are high accurate instruments. Understood? Yes. So what is stethoscope? It is used to, a stethoscope is used to detect the sound produced by the internal organs of human body. Then it is used to identify and localize health problems and diagnose disease. Have you all understood? Okay. Our next topic is sphygmomanometer. This is a clinical instrument. Sphygmomanometer is a clinical instrument. It is used to measure blood pressure. The function is used to measure the blood pressure of a person. Yes. So these are the two sphygmomanometers, two types. Now we can see the parts of a sphygmomanometer. Yes. So this is the first type, that is the monometric sphygmomanometer. So the main part is mercury manometer. This is the main part, this full area, this is mercury manometer. The next part is BP cuff or blood pressure cuff. Then third one is inflation bulb. This is the inflation bulb. This bulb only zoomed here, inflation bulb. Now mercury. So at the bottom of this mercury manometer, one mercury ball is present. Yes, because of that mercury ball only we can find out the pressure, blood pressure. The ball, because of that, this manometer, that readings rising. Yes, the last one, box case, the white color area, outer area, that is a box case. So these are the parts. Once again we can go through this: mercury manometer, BP cuff, inflation bulb, mercury and box case. So these are the parts are present in the sphygmomanometer. Yes, this is a old type of sphygmomanometer. Now this sphygmomanometer, that is a Greek word, so it is derived from two Greek words, that is sphygmos and manometer. Sphygmos meaning heartbeat and manometer is device. So sphygmos plus manometer, sphygmomanometer, it is used to find out the blood pressure. So find out the blood pressure, that is a use, sphygmomanometer function or use. That is a Greek word. So these are the two words derived from Greek word, sphygmos plus manometer. Now we can see who discovered this sphygmomanometer. So he is an Italian doctor. His name is Scipione Rivarocci. Scipione Rivarocci in 1896. So he has discovered sphygmomanometer. He is an Italian doctor. So that doctor only found the sphygmomanometer. Scipione Rivarocci. So this is a sphygmomanometer, that is a full apparatus. Now here we can see the doctor and one patient. So what is she doing? She is finding the BP of this particular patient. Yes, so through the sphygmomanometer she can estimate the state of blood circulation and working of the heart and increased or decreased blood pressure. So these two areas she can identify. So what are the two areas? Estimate the state of blood circulation and working of the heart, then increased BP or decreased BP. So these all the doctors can identify with the help of sphygmomanometer. Yes, so decreased BP or increased BP, both they can identify. Now the normal BP is 120 by 80 mmHg. That is the normal blood pressure, 120 by 80. 120 is systolic pressure and 80 is diastolic pressure. 120 is systolic pressure and 80 is the diastolic pressure. Now this is also another one sphygmomanometer, that is the modern digital type of sphygmomanometer. Modern digital type. So doctors are using this type also. So both the types are available in the hospitals, old one as well as new one. Both the types are available in the hospitals. Both can show the correct readings, that is high BP or low BP or high BP. Both through the instruments doctors can identify. So this is digital one. Now we can see the blood pressure categories. So blood pressure category, these are the 5 blood pressure categories. So first category is normal, second one elevated, third one high blood pressure stage 1, next one high blood pressure stage 2, and the last one is hypertensive crisis. So normal means less than 120. Systolic BP is less than 120 and diastolic level is less than 80. That is the normal blood pressure, 120 by 80. Then elevated, that is 120 to 129 and less than 80. So this also related to normal only. Because of this BP also there is no problem. So 120 to 129 and diastolic level is less than 80. That is the elevated conditions. Then high blood pressure stage 1. That is the systolic BP is 130 to 139 and diastolic level is 80 to 89. Then high blood pressure stage 2, 140 or higher and the diastolic level is 90 or higher. So these two areas, when they are feeling high BP, immediately they have to consult with the doctor and have to take the proper treatment also. Then the last one is the dangerous condition, hypertensive crisis. That is higher than 180 and here higher than 120. That is the dangerous conditions. Immediately the particular person have to meet the doctor and they have to get the treatment also. Yes, so these are the blood pressure categories. Anyway, normal is less than 120 and less than 80 or 120 by 80. That is the normal BP of a particular person or healthy person. Have you understood? Exercise number 2. Question number 1. What are heart sounds? How are they produced? What are heart sounds? How are they produced? Question number 2. The complete events of cardiac cycle last for 0.8 seconds. What is the timing for each event? The complete events of cardiac cycle last for 0.8 seconds. What is the timing for each event? Question number 3. Doctors use stethoscope to hear the sound of heart. Why? Doctors use stethoscope to hear the sound of heart. Why? Number 4. Define pulse. Define pulse. Number 5. Define blood pressure. Define blood pressure. Number 6. Define cardiac cycle. Define cardiac cycle. Blood groups. The concept of blood grouping was developed by Carl Landsteiner. The blood grouping was developed by Carl Landsteiner. He identified blood groups A, B and O. 3 groups he has identified, A, B and O. AB blood group was recognized by De Castello and Steny. Another one blood group, that is AB. This was identified by De Castello and Steny. Now, human blood contains specific substances. The human blood contains specific substances. They are known as antigen and antibody. So, these are specific substances or protein. That is a specific protein. So, this antigen is present in the surface of RBC and the antibody is present in the plasma. So, antigen is present in the surface of RBC and antibody is present in the plasma. Based on the presence or absence of antigen and antibodies, human blood group is classified into 4 groups. So, based on the presence of antigen and antibody, the human blood group is classified into 4 groups. They are A, B, AB and O. 4 blood groups, A, B, AB and O. Now, we have to see. This is the A group individuals, A blood group A. So, this A group individuals, surface of the RBC, antigen A is present. This is the surface of RBC. So, antigen A is present. B group person, surface of RBC, antigen B is present. And AB group, so that person's surface of the RBC, both the antigens are present, A antigen and B antigen, both the antigens are present. But in O group persons, surface of the RBC, there is no antigens. Here, see, nothing is there, so there is no antigens. Okay, so A blood group persons, RBC, antigen A is present, B group, antigen B is present, AB group, antigen AB is present, both are present, but here O group, there is no antigen. See, A group person, antigen A is present, but antibody B is present. Antibody also protein, special type of protein, but A group person's blood antibody B is present. Then B group persons. B group persons, they are having antigen B and antibody A. Antibody A. Antigen B and antibody A. Now, AB group, antigen A and B both are present, antibodies absent, there is no antibodies. O group, no antigen and antibodies A and B, both the antibodies are present, no antigen but both the antibodies are present. Now, we have to see blood donation. In blood transfusion, one must consider the antigen and antibody matching between the donor and the person receiving blood. So, while transfusing the blood, the person must consider the antigen and antibody matching between the donor and the person receiving blood. So when an individual receives the mismatched blood group from the donor, agglutination, that means clumping of blood occurs in the body which leads to death. So one who is receiving the blood, before that have to check the blood, or mismatched blood should not get. Only the matched blood only have to get, otherwise agglutination occurs which leads to death. So the person is donating blood, that person is known as donor. One who is receiving blood, that person is known as recipient. Now AB blood group, that blood group is called universal recipient. So AB blood group is called universal recipient. Why it is called universal recipient? They can receive blood from persons with any blood group. They can receive blood from persons with any blood group. So they are known as universal recipient. But the O group is known as universal donor. Universal donor. They can donate blood to persons with any blood group. They can donate to any blood group. So they are known as universal donor. Now have to see RH factor. So this RH factor was discovered by Landsteiner and Weiner in 1940 in rhesus monkey. Landsteiner and Weiner in 1940 in rhesus monkey. This rhesus factor is an inherited protein found on the surface of red blood cells. If your blood has the protein, you are RH positive. If your blood lacks the protein, you are RH negative. RH positive is the most common blood type. A baby can inherit the RH factor from either parent. So RH factor is the inherited protein only. So now we have to see one video. So through that, who can donate the blood and who can receive the blood, that we are going to see. This is human body. So within our body there is a system called lymphatic system. So within our body there is a system. That system is known as lymphatic system. You know about organ systems, for example reproductive system, digestive system, circulatory system, like that. This is also a type of system, that is the lymphatic system. So this lymphatic system is comprised of capillaries, vessels, nodes and ducts. It is made up of capillaries, vessels, nodes and ducts. So these capillaries we can say it is lymphatic capillaries, vessels we can say it is lymphatic vessels, nodes lymphatic nodes and ducts we can say it is lymphatic ducts. Yes, so these all the items comprised together and form the lymphatic system. Now we can see the functions of lymphatic system. It removes the unwanted toxins, then waste, remove the waste and remove excess of water. So it removes unwanted toxins, eliminate the waste and excess water. So this is the lymphatic system. This lymphatic system accompanied with the blood circulation, or it accompanied with the circulatory system. Lymphatic system is accompanied with the circulatory system. Go through the picture. Lymphatic system. So here lymphatic vessels. What is lymphatic vessels? Already I told lymphatic capillaries, vessels, nodes and ducts. These lymphatic capillaries unite to form lymphatic vessels. So here this is the lymphatic capillaries. This is another one capillary, this is another one capillary. So these lymphatic capillaries unite to form lymphatic vessels. So what is lymphatic vessels? The lymphatic capillaries unite to form lymphatic vessels. Understand? Yes. Now along these lymphatic vessels one fluid flows. That fluid is known as lymph. Yes. Along with the lymphatic vessels one fluid flows. That fluid is known as lymph. Lymph is a colorless fluid. It is a colorless fluid like water. It is a colorless fluid. So inside the lymphatic vessels, this is a lymphatic vessel. So inside the vessel this fluid flows. Lymph is a fluid. That fluid flows inside the lymphatic vessels. Now this lymph has three main functions. The first function is infection. For example, lymphocytes. You know about lymphocytes, is it? So inside of the lymph or this fluid contain lymphocytes. The lymph fluid contain lymphocytes. This fluid or this lymphocytes fight with the foreign particles or microorganisms. So the particular body or the human body cannot get the diseases easily. Why? These lymphocytes swallow the microorganisms, so easily the diseases cannot affect the human beings. That is the first function. Second one is it remove or it takes excess tissue fluid. Second one, it eliminate the lymph eliminate the excess tissue fluid. The third function is eliminate the waste products, eliminate the wastes. So these are the three functions of lymph. So first function is, what's the first one? It fights with the microorganisms. Why? The lymph contains lymphocytes. These lymphocytes fight with the foreign particles, so microorganisms cannot enter inside the human body. Second one, eliminate the excess tissue fluid. Third one, eliminate all the waste particles, so or waste products. So these are the three main functions of lymph. Difference between blood and lymph. Difference between blood and lymph. Blood is red in color. First point, blood is red in color. Lymph is colorless. Number 2, blood contains plasma, RBC, WBC and platelets. Blood contains plasma, RBC, WBC and platelets. Lymph contains oxygen, carbon dioxide, water, WBC and small amount of nutrients. Lymph contains oxygen, carbon dioxide, water, WBC and small amount of nutrients. Last point, blood flows through veins, capillaries and arteries. Blood flows through veins, capillaries and arteries. Lymph flows through lymphatic capillaries. Once again, listen. Blood is red in color. Lymph is colorless. Blood contains plasma, RBC, WBC and platelets. Lymph contains oxygen, carbon dioxide, water, WBC and small amount of nutrients. Blood flows through veins, capillaries and arteries. Lymph flows through lymphatic capillaries. Exercise number 3. Question number 1. Persons with AB blood group are called universal recipient. Why? Persons with AB blood group are called universal recipient. Why? Number 2. Write any 3 functions of lymph. Number 3. Write the differences between blood and lymph. Write the differences between blood and lymph. Number 4. Who discovered RH factor? Who discovered RH factor? Number 5. Which blood group is universal donor? Which blood group is universal donor?